Okay, so now I want to just go through examples of some of the resources that are available out there for doing protein functional annotation and classification, as well as just mention some of the resources that have been used to generate some of the protein signatures that I spoke about previously. So PFAM is quite a popular resource, and it basically is a resource that contains a collection of profiles used to identify, or profiles generated from different protein families and domains. So remember, the profiles I mentioned before is one of the types of protein signatures. And these profiles have been generated using multiple sequence alignments, and in particular at PFAM, these have been generated using hidden Markov models. So you have profiles representing different protein families and domains. So again, PFAM has two components. And this is related to the level of curation. So PFAM A entries. So these are profiles of high quality that are manually created. And then we have PFAM B entries, and these are automatically generated from multiple sequence alignments. You also have generation of a higher level grouping of related families known as clans, which I haven't spoke about in detail. But if you are interested in looking at that, you can have a look at the documentation on the PFAM website. The next tool is a tool called SMART. And SMART is essentially used, SMART is a resource where they have generated profiles for identifying protein domains. It's a tool that has gone into quite a bit of detail on understanding protein domain architecture, and they also have manually created models for the prediction of various different types of protein domains. The next tool is called PRINCE, and PRINCE now is related to a tool that's used for identifying fingerprints. So it's a collection of protein family fingerprints, and remember, fingerprints are a group of conserved motifs used to characterize a protein family. So we have the prediction of functional families. We can use PRINCE to predict the functional families in uncharacterized protein sequences based on the identification of these fingerprints. Okay, and then another very popular resource is a resource called Xphasy. So Xphasy is an online resource hosted by the Swiss Institute of Bioinformatics. And some of you might have come across it before. It isn't only used for protein bioinformatics. It's actually a whole collection of tools, databases that can be used for any type of sequence DNA analysis. They do have a section that is devoted mostly to protein sequence analysis. And in addition to tools used for protein sequence analysis, they also have a variety of tools that can be used for analyzing protein structural data. I think in the practical session, you'll have a look at this resource as well. Then there's a protein information resource. So this is a more general resource that contains information on protein ontologies, reports directly generated from UniProt KB. So these are just general reports on protein information for different proteins. And they also have tools for doing literature mining and text mining. Then the next resource that I want to speak about, so just to recap what I said before, is that there are a variety of different databases. There's a variety of different resources. And I spoke about some of the resources like PRINCE and one of the other ones used for generating protein signatures. Okay, and it can be quite difficult trying to remember what is where and to be able to access all that information from the different resources. So like I said before, there's a number of resources that have been developed to allow you to integrate all the information together. And one of those resources is a resource called InterPro, and it's designed to integrate information from the different signature databases. So essentially what you can do in InterPro is you can submit a sequence of interest. So you might have a protein sequence for which you do not know the function. And what InterPro will do is it will run all the different protein signature approaches on that sequence of interest and give you back all the information that it can possibly annotate on that sequence using the different protein signature approaches. So it is a good sort of one-stop shop that you can use in order to find, so you can use it to find information on any protein sequence, and you can use it to try and annotate any protein of sequence of interest that you have. So this is just a graphical overview of all the different resources that InterPro actually uses and runs when you run an analysis. So it pulls in pattern information from ProCite, it pulls in profile information from these various resources, it pulls in fingerprint information from prints, and then it pulls in protein family information and domain information from these various resources, and then it also pulls in protein structural information from these resources. So you can see that it's a good one-stop shop to get information on anything for a particular protein. And so what they have developed is an InterPro scan tool. So this is essentially the tool I was talking about before, where you're able to submit a sequence. It'll then run all the protein signature methods against that sequence and give you back a results page with all that information, with the signatures that match on your protein sequence of interest. So we're not going to be having a look directly at InterPro scan. In the practical, we're going to be using UniProt. But I would suggest that you actually go and have a look at InterPro scan and run, we're going to be using a sequence of interest in the practical, run that same sequence using InterPro scan and have a look at the results that you get back. Okay, so finally what I want to do is I want to go through an entry in the UniProt database, or the UniProt knowledge base, and show you what type of annotation information you can receive back for a particular protein of interest. So in this example, I'm using the PAX6 protein from humans. So I want to show you what information you can get back. So if you search for the PAX6 protein in UniProt, this is the results page that you'll get back, and I've just sort of broken it up into the different sections on each one of the slides. So first thing that you'll see is you'll get back a results page telling you the name of the protein, what gene is it encoded from, what organism, and then you'll get a status line. So remember, the status line is going to tell you to what level has this protein sequence been reviewed. And in the case of this sequence, it has been completely reviewed. You'll see it gives an annotation score, which is full, and it tells you that there is experimental evidence at the protein level for this existence of this particular protein. It then will tell you something about function. So it will tell you what is the known function of this particular protein of interest. And in this case, the PAX6 gene is a transcription factor, and it gives you some more information on that. It then will tell you some information about the domains. And again, you'll see here that it says bisimilarity. So that means that the function of this protein has been determined based on comparison with other proteins of known function and other proteins with domains in which the function is known. And then on the results page that you get back, this is in the left-hand panel, and these are the different sections and information that you get back for this protein sequence. So I'm just going to go through some of these very quickly and show you what information you get back. So if you scroll down further on the page, you'll find a section on gene ontology. So this will tell you, first of all, you'll see, or before you get to that, it will show you there's a region section. And basically what this will do is it will show you if there's any key features that have been identified on the sequence. And in this case, there is a DNA binding domain. And this is, the description is, it's a homeo box, and it's telling you where it's pulling that information. It's actually pulling that information from the ProCite database. And this is sort of a graphical overview of the sequence. So it's showing you graphically in the whole protein sequence where is this domain that has been identified. You then have some information on gene ontology. So it tells you what is the possible molecular function of this protein, what molecular function is involved in, and then what biological process is involved in. You then have names and taxonomy, so it'll tell you whether this protein is known by any other name. It then gives you link outs to external databases. For where that information, where you can get further information on that protein. It also has a subcellular location section, so where is this protein known to be expressed and translated. There's a pathology and biotech section, so this will tell you if mutations in this gene are known to be involved in any disease. It then also gives you those particular mutations. It gives you link outs to the publications that have identified those mutations, and then it gives you a graphical overview of where those mutations are with respect to the actual amino acid sequence. You then have a section on post-translational modification and processing. So is there any evidence that this protein sequence is post-translationally modified? And it tells you here that there is some evidence that the sequence is ubiquitinated by TRIM11. And again, it is giving you resources that you can go to find information to validate those interactions, or that post-translational modification. There's a section then on expression, so where is this protein known to be expressed? And it will tell you if it's organ-specific, and again, you have link outs to the database supporting that information. You then have an interaction section, so is there any evidence for known interaction between this protein and any other protein? So again, we saw before that TRIM11 does interact with this protein, and again, it will tell you if there are any proteins that interact with this protein that we're looking at part6. There then is also a structure section. So again, we haven't looked at protein structure in this section of the course, but essentially a database where all protein structure information is stored is a database called PDB. So if you are looking for any information on a protein structure, PDB is where you need to look for that. So this will give you some information about the secondary structure. So what is the section? What is the composition of the secondary structure in terms of helices, turns, and beta strands? And then you have link outs to additional information about the actual 3D structure for this protein sequence if it has been solved. We then have a section on family and domains and repeats. So again, it will tell you some more information about particular domains that have been identified in this protein, as well as where it's pulling the information from. And you can then look at more detail about what domains have been identified in your protein. You then have additional link outs to family and domain databases. So again, you'll see some are familiar here, we've spoken about before: InterPro, PFAM, Print, Smart, ProSite. And then these are individual accession numbers that allow you to link out to those individual databases. And you'll see that there should be consistency in terms of the domains that have been found, as well as the family to which this particular protein belongs. So in this case, you'll see that this homeobox domain has been identified, and you'll see that it's been identified across a number of different resources that have been using different protein signature approaches to identify those domains. So you can actually click on these to link out to the individual databases. You then have a sequences section, which will obviously give you the sequence of the protein. And then you have a cross-references section, which will give you link outs to the primary information that's been used to generate the information on this page. So you have link outs to EMBL, GenBank, and DDBJ, and link outs to RefSec, etc. Okay, so I think that's all I want to speak about in this lecture, and hopefully you will enjoy the practical session and you'll get to play around and explore some of these resources in a bit more detail.